Welcome! We're going to explore the topic of pharmacokinetics, which simply means what the body does to a drug. Whenever you take a medicine, it doesn't just magically work. It goes on a journey through your body. That journey is described by four main processes: absorption, distribution, metabolism, and excretion, or ADME for short. Let's break it down step by step. For most drugs to work inside your body, they must be present in the bloodstream. Absorption is how a drug enters the bloodstream. Most drugs are taken orally, so the process starts when the drug reaches your stomach, where it dissolves or disperses. It then transits to the small intestine, where most drug is taken up into blood. Many factors affect how fast or how well a drug is absorbed. Absorption is affected by the drug's formulation. So, is it in pill or tablet form? Is it in a capsule? Or is it a liquid medicine? Absorption also depends on the chemical properties of the drug. For example, is it fat soluble or water soluble? The presence of food can affect absorption. So, was the drug taken with food or on an empty stomach? The environment also affects absorption. For example, its pH. This is important when you consider that the stomach is highly acidic, whereas the intestines are alkaline. The stomach is a minor site of absorption for most drugs. It's lined with thick mucus to prevent damage from its own acid, and that acts as an extra barrier between drugs and blood. Compared with the rest of the gastrointestinal tract, the stomach has a relatively small surface area, further limiting the opportunities for drugs to access the circulation. Also, drugs spend a relatively brief time in the stomach before moving on to the intestines. An important feature of the stomach is its high acidity, which helps it to break down food and kill microorganisms. Some drugs need the acid environment to dissolve well. For example, iron supplements, some antifungal and HIV drugs. On the other hand, acid can also break down drugs, destroying them even before they can be absorbed. Protein drugs like insulin and biologics are examples of drugs that don't make it past the stomach when given orally. Some penicillins are also affected. The stomach contains digestive enzymes, which also break down protein-based drugs. Absorption is affected by delays in the stomach. Many factors affect the rate at which the stomach empties, and therefore how long it takes a drug to get out of the stomach and reach the intestines. One of these factors is food. Both the amount of food in the stomach and the type of food can affect the rate of stomach emptying. In addition, food can change how a drug dissolves and can even bind to it. Several pharmaceutical strategies protect drugs from stomach pH. One approach is to avoid the stomach altogether by administering the drug via an alternative route. Drugs given by intravenous injection go straight into the bloodstream and avoid absorption altogether. Drugs delivered by subcutaneous injection are gradually absorbed from the tissue via local capillaries. Drugs can also be inhaled into the lungs, from where they are absorbed via capillaries next to alveoli. Orally administered tablets can be manufactured with an enteric coating, which doesn't dissolve in acid and protects the drug until it reaches the small intestine. Examples include enteric-coated aspirin and the proton pump inhibitor omeprazole. Microencapsulation is another approach. It involves surrounding tiny beads containing the drug with a protective coating and enclosing them in a capsule that dissolves when it reaches the stomach. Omeprazole capsules are prepared this way. Another approach is to chemically modify the drug molecule to improve its stability in the stomach. That could involve making a salt form that retains pharmacological activity, or a prodrug that is acid insensitive but broken down in the body to the active molecule. Enalapril is a prodrug that is converted to the ACE inhibitor enalaprilat in the liver. Although not usually the best solution, agents that buffer pH can be given with a drug to help it survive the stomach. An example is the use of antacids with antibiotics. Most drug absorption occurs in the small intestine. Stretching several metres in length, it's the longest section of the gastrointestinal tract and offers an enormous surface area for drugs to enter the circulation. The prolonged transit time through the small intestine further enhances the opportunity for absorption. The intestinal wall is highly specialised for nutrient uptake, with its luminal surface covered in villi and microvilli, increasing the surface area further. This cartoon illustrates the intestinal wall. The lumen, where drug is delivered, is shown at the top, while the outer wall lies at the bottom. The finger-like projections covering the luminal surface are the villi, and each projection is a villus. A single layer of epithelial cells surrounds each villus. On closer inspection, each epithelial cell is itself coated with tiny extensions known as microvilli, dramatically increasing the surface area for absorption. Each villus extending into the lumen contains a rich network of capillaries. To be absorbed into the bloodstream, a drug must first cross the epithelial barrier and then enter the capillary network within the villi. The most common method for drug molecules to cross the intestinal epithelium is passive diffusion. In this process, drugs move down their concentration gradient from an area of high concentration in the gut to low concentration in the villi. Lipophilic, fat-soluble drugs pass easily through cell membranes, making this route the most efficient for small, non-polar and lipophilic drugs. In contrast, hydrophilic, water-soluble drugs do not readily diffuse across membranes. Some drugs rely on facilitated diffusion to cross the epithelial membranes. This process involves specialised carrier proteins embedded in the cell membrane. It does not require energy, but depends on the presence of the appropriate transporter, meaning that only drugs that can bind to available carriers are able to use this route. An example is gabapentin using the L-amino acid transporter. A few drugs hitch a ride on active transport systems. These are membrane-bound transporters that use energy in the form of ATP to move molecules against their concentration gradient. This method is important for drugs that resemble nutrients. For instance, levodopa, which is structurally similar to tyrosine, uses the same amino acid transporters. In rare cases, very large molecules can be transported by pinocytosis. In this process, the cell membrane engulfs the drug into a vesicle, which then travels through the cell and releases its contents by fusing with the opposite membrane. Though not commonly used for drug absorption, this route is crucial for the transport of protein-based drugs and nanoparticles. Drug distribution is how drugs move in the body on their journey from the bloodstream to their targets in the tissues and organs. The efficacy or toxicity of a drug depends on its distribution in specific tissues. Drug transfers reversibly between blood and the various tissues of the body, but The rate and extent of transfer depend on blood flow to the tissue and the physicochemical properties of the drug. Blood flow to your organs varies with their metabolic needs, and some organs are more metabolically active than others. Areas of high blood flow are the brain, heart, liver, and kidneys. Areas with the lowest flow are the skin, fat tissue, and bone. Higher flow helps a drug to quickly and efficiently reach its target organs. The physicochemical properties of a drug that influence where it goes and how widely it spreads are its molecular size, its lipophilicity, its state of ionization at physiological pH, and the extent to which it binds to proteins in blood plasma. To reach tissues, a drug must cross the capillary wall, which is formed by a monolayer of endothelial cells. Drugs can pass between cells, known as the paracellular route, or through cells, known as the transcellular route. Small molecules move more easily between or through endothelial cells than large molecules. Transport is also affected by the relative solubility of a molecule in the lipid membrane or watery blood. Lipophilic drugs cross easily by either route, so distribute widely into tissues with a rich blood supply. In contrast, hydrophilic drugs stay mostly in the blood unless there is a transporter protein to assist. Most drugs are weak acids or weak bases, meaning that they are partly ionized in solution, with pH determining the degree of ionization. Drugs that are mainly non-ionized or neutral at blood pH cross the endothelial membrane easily, whereas drugs that are mostly ionized or charged can remain trapped in the blood. The ability of a drug to move from blood into tissues is also affected by its affinity for plasma proteins, mainly albumin. Many drugs bind reversibly to albumin, and that holds them in the blood because only the free drug can cross the endothelial barrier to the tissues. A classic example of a drug that is highly plasma protein bound is warfarin. With 97 to 99% of it bound, only 1 to 3% is available to cross into tissues and exert its therapeutic effect. Bringing this all together, small size, lipophilicity, non-ionization, and low plasma protein binding favour wide distribution. In contrast, large size, hydrophilicity, ionization, and high protein binding restrict distribution. An estimate of how drugs distribute in the body is obtained by calculating the volume of distribution, abbreviated to VD. It's more correctly known as an apparent volume of distribution because it is a theoretical volume, not an actual volume. It's defined as the volume in which a drug appears to be distributed throughout the body relative to blood plasma. It is calculated using the formula VD equals the amount of drug in the body divided by the concentration of the drug in plasma. The amount of drug in the body being the dose administered. The units of the calculated value can be worked out as an amount, usually in milligrams, divided by concentration in milligrams per litre. The amounts cancel out, so the unit is 1 divided by 1 divided by litre, which becomes litre. Let's look again at the endothelial barrier separating blood from tissues. To measure volume of distribution, drug is injected directly into blood to bypass absorption. If a drug mostly stays in the blood, at equilibrium, very few molecules will have crossed into the tissues and the volume of distribution is low. On the other hand, if the drug easily crosses into tissues and only a small amount remains in blood, we see a high volume of distribution. So, a high VD means that drug distributes widely to tissues, lipophilic drugs, for example. A low VD means that the drug remains mostly in the bloodstream, for example, hydrophilic drugs. The volume of distribution is a useful measure for understanding where a drug distributes, predicting how long it stays in the body, calculating doses, and adjusting treatment for patient conditions. While drugs distribute to most tissues according to their physicochemical properties and blood flow, some barriers significantly restrict drug movement. A key example is the blood-brain barrier. As elsewhere, drugs have to move across the endothelial cells lining brain capillaries. But the endothelial cells in the brain have tighter connections, making it harder for drugs to cross by the paracellular route. The barrier is further reinforced by astrocyte end feet that completely surround the endothelial cells. The function of the blood-brain barrier is to protect the brain from potentially harmful substances in blood. But it means that drugs able to freely distribute into other tissues may have difficulty penetrating the brain. Body fat can impact drug distribution. It increases the volume of distribution of lipophilic drugs, which accumulate in fat. Body water has the same effect on hydrophilic drugs. So the excess tissue fluid in edema provides more space for hydrophilic drugs to distribute into. Age is another factor, especially in newborns, which have a higher water content than other age groups. The liver is the main producer of plasma proteins, and the kidney is important for retaining them. So, liver and kidney disease can alter plasma protein levels and affect the amount of drug that is bound by plasma proteins. Drug metabolism is how the body chemically alters drugs. The purpose of metabolism is to make drugs more amenable to being removed from the body. The effect is to make the drug more water-soluble. That usually results in the drug becoming inactivated, but in some cases the drug is activated by metabolism. A drug that is activated in this way is known as a prodrug. The liver is the major site of drug metabolism, but the intestines, kidney, lungs, plasma, skin, and brain can also contribute. Drug metabolism employs enzymes that are located mainly in the liver. They metabolize drugs in two phases, each of which serves a particular purpose. Phase 1 reactions modify the drug to introduce or uncover a functional group. That prepares it for phase 2 reactions, which conjugate the drug by attaching a large polar water-soluble group. You can think of phase 1 as tweaking the drug to prepare it for phase 2, which then bolts on a handle for its elimination. These reactions usually occur simultaneously, but depending on a drug's structure, it may require only phase 1 or only phase 2 to be ready for excretion. Let's look first at phase 1 reactions, which create or uncover a chemically reactive functional group in a drug molecule. The types of reaction that add functional groups are oxidation and reduction. The reactions that uncover functional groups are hydrolysis and hydration. Both are known as functionalization, and these functional groups are commonly involved. The effect in each case is to make the drug slightly more polar, so slightly more water-soluble. Phase 1 reactions are the main reactions that activate prodrugs. The most common type of phase 1 metabolism is oxidation. It is mediated by cytochrome P450 enzymes, abbreviated to CYPs. They are a large superfamily of heme proteins with a variety of functions. However, the CYP1 to 3 families account for the majority of phase 1 metabolism of small drugs. In fact, the CYP3A4 isoform accounts for half of all reactions. A key feature of CYP enzymes is that their activity can be modulated by drugs, as well as substances commonly found in grocery stores or health food shops. For example, furanocoumarins in grapefruit juice inhibit CYP3A4, slowing down drug metabolism and potentially leading to toxic levels of medications even when taken at the prescribed dose. In contrast, St. John's wort, a popular dietary supplement, induces CYP3A4, accelerating metabolism and thereby lowering drug concentrations below therapeutic levels. The interaction between alcohol and CYP enzymes is more complex. Acute alcohol consumption inhibits the activity of several CYPs, while chronic use induces some CYPs. However, long-term alcohol use also causes liver damage, which can impair overall drug metabolism and complicate dosing further. Phase 2 reactions involve conjugation, meaning that a polar molecule is attached to either the active drug or its phase 1 metabolites. There are many types of conjugation reaction, such as glucuronidation, sulfation, etc. Each reaction involves specific transferase enzymes that catalyze the transfer of the polar group to the drug. The product of conjugation is larger than the original drug molecule and more water-soluble, making it easier to excrete. Adding a bulky group onto a drug usually inactivates it by reducing its affinity for its target. Phase 2 reactions mainly take place in the liver. There is a caveat to what I told you earlier about drugs being absorbed into the circulation from the intestine. Instead of entering the systemic circulation directly, drugs absorbed from the intestine enter the mesenteric veins, then travel via the hepatic portal vein to the liver. There, they may undergo metabolic transformation before even reaching their target sites. As a result, only a portion of the administered dose reaches the systemic circulation in its active form, a proportion referred to as the drug's bioavailability. Unmetabolized drug leaves the liver via the hepatic vein to join the venous blood returning to the heart. From there, it circulates to its site of action. The bioavailability of a drug is measured by determining how much of it reaches the systemic circulation in its active form compared to when the same dose is administered intravenously. As intravenous administration delivers the drug directly into the bloodstream, it is 100% bioavailable. To measure bioavailability, volunteers are given a fixed dose of the drug by the test route or by the intravenous route. In our example, the test route is oral. Blood samples are collected at multiple time points after drug administration, and the plasma concentration of the drug in each sample measured. The values are plotted on a graph of plasma drug concentration versus time. The points are connected, and the area under the curve for each data set calculated. The area under the curve is shown here for the intravenous route, and now for oral administration. Bioavailability is then calculated as the area under the curve for the oral route divided by the area under the curve for intravenous drug. The result is a dimensionless ratio. Knowing the bioavailability of a drug is important because it directs how it is formulated, administered, and dosed. The final stage of a drug's journey through the body is its excretion. The most common route for drug excretion is via the kidneys and urine. The liver is another major route directing drugs through bile to feces. Although minor, additional routes for drug excretion are the lungs, saliva, sweat, tears, and breast milk. While saliva, sweat, and tears play a minimal role, the lungs are an important route for volatile anesthetics and alcohol, and drug accumulation in breast milk is an important consideration in breastfeeding. This cartoon depicts a nephron in the kidney. Drug travelling in the bloodstream reaches a network of capillaries known as the glomerulus. Blood is filtered under pressure here, known as glomerular filtration. Small molecules, including drugs, pass from the blood into the Bowman's capsule at the head of the nephron. Drugs bound to plasma proteins are too large to pass through the glomerular membrane, and they remain in blood. So, only free, water-soluble, low molecular weight drugs or their metabolites are in the filtrate, which enters and travels along the proximal tubule. Organic ion transporters actively transport additional drug molecules from blood in the capillaries into the proximal tubule. As the fluid continues through the nephron, lipid-soluble drugs can diffuse passively back into blood, while water-soluble drugs remain in the tubules, reaching the collecting duct, then bladder, as urine. The pH of urine can affect renal excretion by determining the ionization state of a drug. Acidic urine favours reabsorption of weak acids, while alkaline urine favours reabsorption of weak bases. The types of drugs that are excreted by the liver are typically large and polar, often conjugated drug metabolites. Drugs are actively transported into bile canaliculi and become part of the bile. Active transport means that the process requires energy in the form of ATP. The transporters involved include a multi-drug resistance associated protein, bile salt export pump, and P-glycoprotein. Bile flows through the bile duct into the gallbladder for storage or directly into the duodenum of the small intestine. It can then pass through the intestine to become excreted in feces. An alternative fate for drugs excreted this way is to become recycled as part of the enterohepatic system. Drug metabolites in the intestines can be deconjugated by gut bacteria using enzymes like beta-glucuronidase. That can reactivate the drug and restore its lipid solubility so that it can be reabsorbed into the mesenteric circulation. The drug returns to the liver via the hepatic portal vein, where it can be metabolized again or enter the systemic circulation. In this way, the cycle can repeat itself over and over again. The advantages of enterohepatic recycling are that the half-life of the drug in blood is prolonged, maintaining therapeutic levels for longer. However, drugs can accumulate in the liver, and it is a source of drug-drug interactions. Drug elimination is the inactivation and removal of a drug from the body and includes both metabolism and excretion. The rate of elimination is measured from plots of plasma concentration versus time, which we used earlier to measure bioavailability. Sometimes the plasma concentration falls at a constant rate over time, giving a linear relationship. This means that the rate of elimination is independent of the concentration of the drug and occurs when drug elimination follows zero-order kinetics. Therapeutic drugs rarely follow zero-order kinetics, but one drug that does is ethanol. That is because the enzymes responsible for metabolizing it become saturated at low ethanol concentrations. The elimination of most drugs follows first-order kinetics. That means that constant fraction of the drug is eliminated per unit time. When plasma concentration is plotted against time, the relationship is exponential. So the rate slows as the concentration decreases. In both cases, the rate of elimination is indicated by the half time, abbreviated to T1/2. To measure this, draw a horizontal line through one of the points on the plot and another line at 50% of the plasma concentration at the point selected. Drop a vertical line down from where the 50% line meets the data. Where it crosses the time axis is the elimination half time. Drug clearance refers to how efficiently a drug is removed from the bloodstream. It can be calculated for the entire body or specifically for organs like the liver or kidneys. Clearance is defined as the volume of plasma from which a drug is completely removed per unit time. The basic formula is clearance equals the rate of elimination divided by the plasma concentration. It is expressed as a volume per unit time, such as milliliters per minute. For a drug that follows first-order kinetics, both the elimination rate and the plasma concentration change over time. To avoid this variability, we use a more stable calculation, especially when the drug is given intravenously, ensuring 100% bioavailability. In this case, clearance equals the dose administered divided by the area under the curve of the plasma concentration versus time plot, which you have seen before. The area under the curve represents total drug exposure and is measured from time 0, when the drug is administered, until it is eliminated from the body. The clearance of a drug is influenced by two key parameters: half-life and volume of distribution, linked by the equation clearance equals 0.693 times the volume of distribution divided by the half-life, where 0.693 is the natural log of 2. This tells us that a large volume of distribution, or short half-life, increases clearance. On the other hand, a small volume of distribution, or long half-life, reduces clearance. Let's finish by bringing everything together with a summary of the pharmacokinetics journey. That is, what the body does to a drug from entry to exit. First, absorption. This is how a drug gets into the bloodstream, usually from the gut. It's influenced by factors like solubility, ionization, and first-pass metabolism in the liver. Next is distribution, how the drug moves around the body. This depends on blood flow, how well it crosses membranes like the blood-brain barrier, and how much binds to plasma proteins like albumin. Then comes metabolism, mainly in the liver, where drugs are chemically altered. Phase 1 reactions prepare the drug structure for Phase 2 reactions, which add groups to make it more water-soluble for excretion. Finally, excretion removes the drug from the body, usually through the kidneys and urine or via bile into the intestines. Some drugs even undergo enterohepatic recycling, returning to the liver for another round. All these processes affect how much drug reaches its target, how long it lasts, and how often it needs to be given. Understanding pharmacokinetics is essential for prescribing safely, designing effective drugs, and predicting how patients will respond.